Okay, so what I was saying was on this slide here, right, if you have normal tissue and these are our differentiated cells, maybe at some point you'd end up with this one cell in there that got some mutational hits. And those are the hits that might initiate the transformation. Then over time, it builds up more and more hits as it divides. And so that it becomes that it's sort of taking over then the population of cells here all represent carrying the same mutational burden as this one initiating cell, but over many generations then it begins to preferentially expand because these cells now have mutations that give them an advantage for dividing out of control. So this whole phase here we might consider precancer. So this may still be, for example, a benign polyp or a tumor forming, which then as they begin to transform further and further, some of them, and that's the important point here, if you look over here, some of them, because they are all rapidly dividing and all unstable genetically, some of them are going to stumble upon the next set of mutations that give them additional advantages. So you can begin to see that those are the really aggressive cells there. And at this point then, by the time this happens, we then end up with truly a cancerous lesion. So CIS refers to cancer in situ. That means cancer in place. At this point, the tumor is still in its original site in the tissue. When it really then becomes cancer is when then these cells, for example, epithelial cells, then gain the mutations to break free of that confinement. For example, if these are epithelial cells, if they finally find a way to break free of the basement membrane and infiltrate or invade the stroma beneath, then that's how we define this as no longer a benign tumor, but is becoming cancerous. So not surprisingly then, depending upon what tissue you're talking about, where this very first cell is starting to go bad or go rogue, right, those cells are already differentiated in such a way that certain pathways in them are emphasized over other pathways. So a cell in your colon might have different signaling pathways in it that govern proliferation that might be different than, for example, the pathways in the epithelial cells of your lungs that govern their proliferation. So depending upon where the original cancer cell starts going bad, you'd have very different kind of progressions of how long it takes for these different cancer sites to actually proceed and become full-blown cancer. So what this shows us is in the colon, typically this whole initiation stage might be 5 to 20 years of just having one of these or a few of these early transforming cells. And then you'd have 6 to 8 years, or sorry, 5 to 15 years in colon cancer, going from just a benign tumor and adenoma to full-blown cancer. Something like lung cancer is a much different kind of etiology or progression, because lung cancer is very much a result of repeated exposure, constant exposure to things like cigarette smoke. So daily load of mutational carcinogenic factors. So as a result, it has a much more gradual, uniform kind of progression from initiation to full-blown cancer. And you can see other cancers as well. Prostate cancer, it might take 20 years of this initiation stage before you have a neoplasia. So this is prostatic intraepithelial neoplasia, not cancer yet, but then another slower drawn out progression until we finally get 3 to 15 years later, full-blown cancer. So this means that cancer progression takes multiple paths depending on what types of cells you're dealing with. And again, each type of cell that's transforming still has to, by chance, just stumble upon the right combination of mutations that takes it to the next step of transforming. And with each one of these steps, the transformation goes faster as additional brakes are released, different tumor suppressor gene pathways are mutated and deactivated, and as different oncogenic pathways are activated improperly because of mutations. So let's look at something, for example, this colon cancer. and this is just an individual case, an example. You start off with normal epithelium. Maybe the first mutational hit is loss of the APC gene, that adenomatous polyposis coli gene that controls beta-catenin. So without beta-catenin being controlled, beta-catenin signaling happens out of control, and that leads to hyperplastic epithelial growth, right? Then maybe sometime later, a second hit happens that has to do with hypomethylation. So for some strange reason, now you begin to have lower methylation patterns within this transforming cell. And as a result, different types of genes that should be turned off then get turned on. That might be followed then through the progression or growth of the benign tumor, stumbling upon maybe this next mutation is activation of RAS, right? So now for some reason, the RAS gene gets a mutation that now increases the activity of RAS signaling. That is then followed by loss of tumor suppressor genes. So a mutation that relieves this particular tumor suppressor gene on chromosome 18q. And importantly, the big one is eventually loss of p53. Now keep in mind, we're not saying that this is one cell moving through all of this, right? We're saying this is one cell that starts off with one hit, and through this whole process, it's dividing and dividing and dividing. And so along the way, most of its daughter cells are going to die, but some of them make it through and keep going. And those become more and more plentiful. So you have more and more opportunities for one of those daughter cells to get this hit, and then maybe another daughter cell that has this first one plus the second one now gets the third one, and so on and so forth. So by the time you finally get ones that stumble upon the loss of p53, right, that really sets the stage now for full-blown cancer and eventually invasion and metastases. If you look at colon cancer, researchers have actually studied which of these steps happen the most frequently. Okay, remember, mutations are all random, so it might seem strange to think that there's actually a preferred, quote unquote, set of mutations that happen in sequence to lead to colon cancer. But remember, what we're talking about is a natural selection process. And the way they've done it is they've actually kind of backtracked or almost like designed a family tree to kind of like a evolutionary cladogram of how resulting colon cancer cells are related to previous generations of transforming colon cancer cells. And what they found is that, for example, the very first hit seems to be, in most colon cancer cells, loss of APC, although any of these other hits are actually, could be possible as well. Then the second step, the most abundant one, is something like the waking up of KRAS, or mutations in PI3 kinase that result in out of control AKT signaling, right? And again, no matter which of these second mutations it gets, it can then stumble upon a third one, which in this case are mutations in things like SMAD, eventually leading then to the most frequent fourth mutation being p53. So it's really interesting to think about this, that even though mutations are random, there are certain combinations of mutations that, depending upon what type of normal cell and normal tissue you started with, would have a preferred kind of path of getting to becoming a malignant and aggressive cancer. So many cancers then have there are many different pathways they could get through in order to go from normal to full-blown metastatic cancer. There are some exceptions of cancers that actually exhibit much more orderly, predictable progression. So pancreatic cancer is a good example of that. This figure here takes us through the abundant or most common mutations that happen in sequence, going from normal pancreatic tissue to invasive pancreatic carcinoma. And you can see that as you progress, you start off with telomere shortening. About 90% of all transforming pancreatic cells, right, KRAS activation is their first mutation that they stumble upon. And the overwhelming abundance, then the second mutation is inactivation of CDK modulators or CDK inhibitors like P16. Then that's followed by p53 inactivation, and then about half of the resulting ones then inactivate SMAD. So because this is so commonly the path that pancreatic cancers go through, this actually means there's an implication that pancreatic cancer is a type of cancer that can actually proceed and progress very quickly, because the overwhelming popularity of these mutations seems to imply then that this is the one sort of path that gets selected for and is preferentially biased for. And that's why pancreatic cancers, oftentimes from the time when the patient is first diagnosed to when they later succumb from the cancer, it can be a very short time. So I'll tell you guys just a brief story. When I was just after college, I was living on the East Coast, and one of my uncles on the West Coast in California was diagnosed with pancreatic cancer. So I remember my mom called me up and told me, oh, Uncle Ken, he was diagnosed with pancreatic cancer. And of course, at the time, I didn't really know that much about cancer. So I said, oh, that's awful. But I was all the way on the East Coast in Washington, D.C., so it's not like I could see Uncle Ken very easily anyhow. And then within 6 months, my mom told me, oh, Uncle Ken died from his cancer, right? And I was just shocked how quickly it could progress. But that's one of the reasons why, unfortunately, pancreatic cancer is one of the most aggressive, fast progressing types of disease. Okay. So if we look at this cartoon then kind of reemphasizes that. If you have a whole bunch of normal differentiated cells on the top here in a tissue in gray, and one of them gets the hit, so an initiating mutation, right? Maybe, for example, KRAS gets hit. Then it's going to undergo a long time of expanding and having most of its daughter cells undergo apoptosis or just eliminated. So for a typical cancer, right, let's say it takes a good 10 years for that initiating mutation to eventually be sort of reflected in a population of cells that took many, many, many generations to get there. Because maybe after 10 years, you have something like 1 million of these cells inside your otherwise normal tissue. And the idea is, because you have like 1 million of them now, it's very likely that one of them will get a second mutation. So this particular one here gets the second hit. Right? And that gives this particular cell an increased proliferation advantage. So it begins to divide more and more. And preferentially then, what you see is that you get a buildup of cells that have both mutations in them now. And this would be the second clonal expansion that happens quicker, right? Because this double mutant cell has an increased mutation rate. And with each additional mutation it builds up, so you can see here now you have a population of cells that have not only both of those mutations, the green and the red, that they carry along as baggage, but now that you have so many of them and they're becoming more and more unstable, it is much more common now that different individual cells will pick up a third, fourth, or fifth mutation that's random and different than its neighbors. And each one of these mutations, whether it's the yellow mutation, the black one, the blue one, orange, etc., gives each of these individual cells here a different type of advantage. And so they will on their own expand to form subpopulations of cells. And they're all now on their own path of choosing these different, what's the next mutation that they'll come across. What that means then is by the time you have a full-blown large cancerous tumor, the mutation rate has increased. It's more rapid now with each generation because the integrity of the genome is becoming less and less well maintained, and you have more types of mistakes that are allowed through more frequently. That also means the resulting tumor at the end might be one large mass that's actually made up of several different populations of cancer cells. And that's what this slide is. So this slide shows you that this is a section, a cross-section of a single solid tumor. But you can see the dotted lines represent the boundaries of what appear to be different subpopulations of tumor cells within this one large tumor. And you can tell that these different subpopulations have different phenotypes. For example, the one in the blue on the left here, right, they seem to be smaller. You can see how tightly packed the nuclei are. These are the little darker purple spots compared to these cells over here that have a lot more what looks like fatty adipose areas inside of them, right? So somehow the mutations in these cells have tweaked them so that they now produce a lot of, I don't know, lipids, or they store a lot of fat or something. These cells over here in red, you can see they are actually beginning to form what looks like vasculature, right? Some sort of early duct type of formation, like they're organizing themselves again as though they are forming the walls of a gland. Keep in mind, the implication here is that all of these cells originated sometime in the past, years and years ago, from one single cell that got that first initiating hit. But through many years of random mutations, you result in a full-blown aggressive tumor like this that's made up of many different phenotypic kind of features. The other thing that notice here is that because we're talking about random mutations that might wake up or turn off different genes, you might have surprising phenotypic changes like these cells that are suddenly deciding to actually form glands. The most extreme case of this are types of solid tumors that we call teratomas. And in teratomas, you may have heard of these before. These are actually really gross, but oftentimes you'll have solid tumors that have things like hair or fragments of bone in them that actually represent the tumor cells getting so messed up that they've actually woken up different pathways that they have no business waking up and turning into something like a tooth or hair, or in extreme cases, they've even found remarkably well-formed eyeballs inside of a tumor, right? So that's both terrifying, that's why they're called teratomas, because they're terrible tumors, but also it's amazing, because it's a good reminder that all of these tumor cells started with the complete human genome in them, right? So even though it might have started as a muscle cell or a bone cell, the reality is this reflects the potential then to actually wake up or become a different type of cell, depending upon how messed up the regulation is.